Okay, so welcome back to this next video in which we are discussing the P2X receptors. Okay, right. So in the previous video, what we did is we saw the categorization of all ligand-gated ion channels that are known within humans into these three different categories: the P2X-like receptors, the glutamate-like receptors, and thirdly, the nicotine-like receptors, also more commonly called the cis-loop ligand-gated ion channel family. Okay, right. We now know that the P2X receptors, all of them, okay, are within a subfamily of this P2X-like receptors, and the entire superfamily here is named after them because they are the main example. Okay, right. So let's now do an in-detailed study of the P2X receptor family then. So, we know therefore that their structure has to be a trimer, okay? So I'll redraw this picture out. So here is our ligand-gated ion channel. Okay, we'll have the pore going down the middle here, and we know that the overall receptor is made up of these three separate subunits all joined together like so. Okay, so they are a trimer. Now, what we want to do again is look at the structure of a single subunit of that trimer, but we're going to do it in a little bit more detail now. Okay, so we're going to pull out one of these subunits and have a look at its membrane spanning topology. So same protocol. What we know is that the amino terminus will be intracellularly. Okay, you've then got a membrane spanning alpha helix. Now, basically on the extracellular loop between the first and the second membrane spanning alpha helix, what is found is that in all P2X receptor subunits, okay, and I'll tell you about the different subunits in a moment, in absolutely all of them you find 10 conserved cysteines on this extracellular loop. Okay, so I hope you understand what I'm telling you. Always in this extracellular loop you find 10 cysteines. Okay? Now, this has led people to hypothesize that these must be very important in the structure, and that potentially what happens is that you get something forming that looks like this. Okay? So the idea is that these cysteines could be involved in the formation of 5 cis loops. Okay, like so, and I haven't done... Yeah, I always manage to do this when drawing the P2X receptor subunits. It starts off really nicely spaced out and then it becomes squished up towards the end. Never mind. Alright, so the idea is that this would be a cis loop here, 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 and this would be a cis loop here. So, the 10 conserved cysteines, therefore, would be at this position here, 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 this one here, this one here, this one here, and this one here. So those are the 10 cysteines. And they've made 5 cis loops. Okay? So, as far as I can tell, there isn't really that strong experimental evidence for this, although people do still sort of draw this whenever they draw a P2X receptor. Basically, if it does look like this, then it doesn't seem that this sort of a structure is important for the function of the subunit, because if you put a reducing agent such as beta-mercaptoethanol or dithiophreinol onto the P2X receptor, which will break all of these disulfide bonds, basically the P2X receptor is still capable of functioning, basically, even once you've broken all of these disulfide bonds. So, until we get the X-ray crystal structure, basically, of this, which I don't think has yet been done, we won't know whether this is the true structure of these receptors. Okay, but you can read about that in reviews and whatnot. But it is still hypothesized that there are these 5 cis loops, like so, in the extracellular loop. And people do, whenever drawing P2X receptor subunits, they generally do draw a structure like this. Okay, right. So we'll fit to that convention and draw this. What is definitely known is that there are the 10 cysteines in the extracellular domain, always. Okay, whether they form 5 cis loops is a little bit more controversial. Okay, right. So, now, let's discuss the different types of P2X receptor subunit then. Okay, so, Basically, what would be absolutely perfectly simple would be if there was just one gene which coded for the P2X receptor subunit, and you just used this gene 3 times, made 3 proteins, stuck those 3 identical proteins together, and the world was nice and easy, and you've got a P2X receptor there. It's more complicated than that in reality. Instead, there are 7 known genes for P2X receptor subunits, okay? And these will have different sequences of organic bases, and therefore they'll code for proteins which have different sequences of amino acids. However, all these different subunits will all share this same membrane-spanning topology, so they'll all have 2 membrane-spanning alpha helices, and their amino and carboxy termini will be on the cytoplasmic side, and in the extracellular loop they have these 10 cysteine residues. Okay, so they are all similar in structure enough that they can all form this sort of a membrane-spanning topology, and they can all form a third of a P2X receptor. Okay, so there is the P2X1 then. There is the P2X2, okay? There is the P2X3 P2X receptor subunit. There is then the P2X4 receptor subunit. So P2X receptor subunits have beautiful, simple naming system. Okay, there is then the P2X5 P2X receptor subunit, the P2X6 P2X receptor subunit, and then finally also the P2X7 receptor subunit. And these all have separate genes producing them. Okay, so they all look like this, and they can all form a third of a P2X receptor. Then what we have to deal with is the fact that they are trimerizing together to make these trimers means that we have a huge number of possibilities now, because we could make homotrimers where we stick 3 identical subunits together. For instance, we could grab P2X1 and we could stick a P2X1 here, a P2X1 here, and a P2X1 here, and then make a trimer which was made out of 3 of the same subunits. That would be called a homotrimer. Alternatively, we could do it more complicatedly. We could use different P2X receptor subunits in different slots, and that would create you what's known as a heterotrimer. Okay? And the heterotrimer, hetero obviously means different, so the subunits within the trimer are different basically. Homo means same, so the subunits within the trimer are the same. So, we know of lots of homotrimers. Okay, so you can form a homotrimer of P2X1. We know that that exists. Okay, so what you do is you put 3 P2X1 subunits together to make a P2X1 homotrimer, and that's a functional P2X receptor. There is the P2X2 homotrimer. There's also the P2X3 homotrimer. Okay, in fact, they all form homotrimers but for one. Okay, so there's the P2X4 homotrimer, the P2X5 homotrimer, and there is finally the P2X7 homotrimer. So there is no homotrimer of P2X6. Okay, so all the others are known to form homotrimers. Okay, so heterotrimers now. Heterotrimers are a little bit more complicated, but some decent examples of heterotrimers which are physiologically important are a heterotrimer consisting of P2X1 subunit, so this one, with P2X5 subunits. Okay, so that's an important one. Now, what is known is that you can have receptors which contain both P2X1 receptor subunits and also P2X5 receptor subunits. What is not known is the stoichiometry of these 2 subunits. Neither is the positions of them known, although that might not be so important when we're only discussing trimers. Okay, so we don't know the stoichiometry, and what I mean by that is we don't know whether you put 2 P2X1 receptor subunits and 1 P2X5 receptor subunit, or whether you put 2 P2X5 receptor subunits and 1 P2X1 receptor subunit. So that's what I mean by we don't know the stoichiometry. What we do know is that you can find receptors which are a mixture of the 2. Okay, and then another example of a heterotrimer which is very physiologically important within pain nociceptors, is P2X 2/3. Okay, so this is a mixture of P2X2 receptor subunits and also P2X3 receptor subunits. Okay, and again, the stoichiometry, as far as I know, isn't known. Okay, right. What is known is that you can mix the two together. Okay, and the heterotrimers consist of a mixture of 2 different types of subunits rather than 3 different types of subunits. Okay, right. So, let's now talk about what the actual ligand for P2X receptors is. So the whole point of them being ligand-gated ion channels is that they're usually closed, but when something binds to them, they actually open. Well, basically, the ligand for P2X receptors is ATP, and this is the ligand for all of them. So ATP, adenosine triphosphate molecules, can bind to these receptors, and then it will cause them to open. Okay, so each one of the subunits will have a ligand binding domain for ATP, and when the ATP binds to that domain, it will trigger changes in the receptor subunit, and when you get ATP bound to all 3 of them, then that will trigger conformational changes in all of them, and that will trigger the receptor, the whole receptor thing, to open, so that you've now got an open pore, basically. Of course, you can imagine that if you just get ATP binding to one of them and not to the other 2, then this one will undergo the conformational change that might, to some extent, open the ion channel, but not as well as it would be if you activated all 3 of them. Okay, so you might have differing levels of opening of the P2X receptor. Okay, so let me just discuss ATP as a signaling molecule then. So, basically, it's becoming more and more apparent that ATP does form a signaling role. Okay, so basically what you can have is cells can have ATP within vesicles. Okay, within storage vesicles, often with other neurotransmitters as well, okay, or other transmitters to keep it more general rather than neurotransmitter, okay, and then what can happen is you can exocytose this vesicle, and then the ATP will be released into the extracellular fluid. It can then go and act on P2X receptors on the next cell or on other cells and trigger them to open. Now, when they open, the main ions that the P2X receptors are permeable to seem to be cations, so sodium ions and potassium ions and calcium ions. Now, we need to think what's actually going to happen then if we open this P2X receptor, and we need to think in terms of what's going to happen in terms of electrical potential difference across the cell membrane. So let's draw our P2X receptor here. Okay, here's its ion channel going down the middle there. Okay, and before the ATP is bound, it's closed. Now, let's discuss the concept of electrical potential difference across the cell membrane. So, electrical potential is this concept from theoretical physics. Okay, basically, it's a mathematical model to help us understand reality. Okay, so the mathematical model is this. You ascribe to every point in 3-dimensional space a real number. Okay, so it can be positive, it can be 0, it can be negative. It can be an integer, it can be a rational, it can be an irrational, it can be a transcendental if you want. It can be whatever real number you like. Okay, it can't be an imaginary number. You are not allowed to go into the bigger number system of the complex numbers. You have to ascribe it one of these symbols within the real numbers system. Okay, so to every point in 3-dimensional space you ascribe a real number, and this is known as the electrical potential. Okay, so in principle, you can have a little man with a machine, okay, which measures this electrical potential, and he can go and stand anywhere in space, and he can get a number on his screen which will tell him the electrical potential at that point. Now, if we consider a cell, okay, so let's let this be a cell. Basically, all of the points inside the cell, at least to a good approximation, are in electrical equilibrium with one another. Okay, so the electrical potential of all the points inside the cell is the same, at least to a good approximation. So we'll call this the electrical potential intracellularly. So big E is for electrical potential. I subscripted like that means intracellularly. So this means the intracellular electrical potential. Similarly, all the points of the extracellular fluid are in electrical equilibrium with one another. Therefore, the electrical potential of any point in the extracellular fluid will be the same. So we can call this the electrical potential, big E, and then we'll subscript it E as well for extracellularly. Now, if we then put this on this diagram down here, this is the extracellular fluid. So we have an electrical potential extracellularly, and we have an electrical potential intracellularly. Basically, these two numbers aren't necessarily the same. It's not necessarily true that the electrical potential of the extracellular fluid is going to be the same as the electrical potential of the intracellular fluid. Okay, so there is an electrical potential difference across the cell membrane, or a voltage across the cell membrane. So when people talk about the electrical potential difference across the cell membrane, what they really should say is the electrical potential difference from extracellular to intracellular. You need really to orient the direction in which you are moving, okay, to completely specify what you mean. However, people intrinsically think that you know what they're talking about, okay? So they assume that you understand that they are talking about going from the extracellular fluid to the intracellular fluid, okay? So often people just say the electrical potential difference across the cell membrane without specifying that they're going from extracellular to intracellular, but you should assume that they mean from extracellular to intracellular. Okay, now, another word then just for the electrical potential difference is voltage. So voltage doesn't mean electrical potential, it means electrical potential difference, a change in electrical potential. Okay, another thing I should say is that the units in which electrical potential, this number that's been ascribed to every point in space, are measured are the volt. Okay, so don't let that confuse you between thinking that electrical potential is the same thing as voltage. Voltage is a change in electrical potential. It's a difference in electrical potential, or an electrical potential difference. So the electrical potential difference from extracellular to intracellular, or the voltage across the cell membrane, what this really means is if the little man started in the extracellular fluid with his machine, and he got that machine to measure the electrical potential in the extracellular fluid, he would get some answer. That's this answer here. If he then moves across the cell membrane into the intracellular compartment, and then his machine measures the electrical potential there, the number will change. It will change to this number. Electrical potential difference from extracellular to intracellular means how much does the number on his machine change when he moves from extracellular to intracellular. So what is the answer there in terms of these numbers here? Well, it's the new electrical potential, okay, the electrical potential intracellularly. So what the new number on his screen minus the old number on his screen, that's how much the number on his screen has changed by. That is what is meant by the electrical potential difference across the cell membrane. And this is usually kept at around negative 65 millivolts, which is the resting electrical potential difference across the cell membrane. Okay, right. So what that means is that the electrical potential within the intracellular compartment of the cell is 65 millivolts lower than the electrical potential within the extracellular compartment of the cell. Okay, right. Now that has important bearing with regards to understanding what's going to happen when we suddenly open this P2X channel here. So here is our P2X channel, and let's say we've doused it in ATP so that it's now open. Now, I've said that they are generally permeable to sodium, potassium, and calcium. Okay, generally the permeability is around the same for sodium and potassium, and then it's slightly lower for calcium, but it's still a good permeability for calcium. It's very, very good for calcium compared to most sodium and potassium channels. Okay, right. So To understand what movements of ions then you're going to get through that P2X channel once it's opened, we need to look at the concentrations of these free ions on either side of the membrane, and we also need to consider the electrical potential difference across the cell membrane and the role that that's going to play. So basically, the sodium concentration extracellularly is around 145 millimolar, whereas the sodium concentration intracellularly is around 12 millimolar. So we've got a concentration gradient of around 12-fold that favours the movement of sodium in, simply because the chance of a sodium ion hitting the ion channel from this side and going into the cell is 12 times the chance that the sodium ion will hit from this side and go out of the cell. In addition, all of these ions are positively charged, including the sodium ion, so positively charged ions always want to go to the area where the electrical potential is lower. Now, I've said that the electrical potential intracellularly is at the moment 65 millivolts lower than the electrical potential extracellularly. So at the moment, it is preferable for the sodium ions to be on the intracellular aspect of this membrane. So the electrical gradient is also favouring the movement of sodium in. So when you open the P2X channel, you're going to get a movement of sodium in because both the concentration gradient and the electrical gradient are working together to move the sodium in. Okay, so for potassium, the concentration of potassium extracellularly is around 4 millimolar, whereas the concentration of potassium intracellularly is around 155 millimolar. Okay, so the concentration gradient is around 40-fold, favouring the movement of potassium out of the cell. Okay, so the concentration gradient says move out, but the electrical gradient says the intracellular compartment has a lower electrical potential than the extracellular compartment by 65 millivolts. Okay, potassium is positively charged. It wants to be in the electrical, in the compartment which has a lower electrical potential, which is currently the intracellular currently the intracellular compartment. So the electrical gradient says to the potassium ions move in. So this is a difficult one. The concentration gradient says move out. The electrical gradient says move in. Who wins, basically, is the question. Basically, this electrical gradient isn't strong enough to completely oppose the concentration gradient. So basically, if I draw this in arrows, the concentration gradient says move out. The electrical gradient then says move in. But the electrical gradient is weaker. Okay, so the net movement out, therefore, is this arrow in blue subtract the arrow in turquoise, which will be an arrow like this. So the electrical gradient opposing the concentration gradient is hugely going to reduce the amount of movement of potassium that you get out, but it's not strong enough to completely stop it. In fact, to completely stop the movement of potassium out, you'd actually have to take the electrical potential difference across the cell membrane down to around negative 85 millivolts, the Nernst potential for potassium. Okay, right. Now, for calcium then, the concentration of calcium extracellularly is around 1.5 millimolar. Okay, whilst the concentration of calcium intracellularly is around 100 nanomolar. Okay, that's a 15,000-fold gradient favouring the movement of calcium in. Okay, now, no electrical potential difference that is at all physiological is ever going to be able to stop calcium moving in when you open a calcium channel. However, in this case, the electrical gradient is actually on the side of the concentration gradient, so of course you're going to get movement of calcium in. Now, you might think, well, that will be a massive great movement of calcium in, but you have to remember, even though the driving force is very high, the actual concentration of calcium that you've got extracellularly is minuscule. It's a lot smaller, around 100 times smaller than the concentration of sodium. So the movement of sodium in is still going to beat the movement of calcium in, simply because sodium's present in such a higher concentration here. So even though the driving force on an individual sodium ion is lower than for calcium ions, just because the concentration's bigger, the amount of sodium you're going to get moving in is still going to beat the calcium. Okay, so overall then, we've analysed that sodium will be coming in, calcium will be coming in, a little bit of potassium will be going out. So the main important effects here are the sodium coming in and the calcium coming in. We can neglect the potassium that might leave. Okay, so the main message here is that we are bringing positively charged particles into the cytoplasm of the cell. We're moving them out of the extracellular fluid and dumping them into the cytoplasm of the cell. Now, what effect is that going to have on the electrical potential difference across the cell membrane? Okay, so basically, where you have positive charge, that raises the electrical potential of that area. So if we're bringing positive charge into the intracellular compartment, what that's going to do is it's going to raise the electrical potential of the intracellular compartment. So the electrical potential intracellularly is going to go up. Meanwhile, we are removing positive charge from the extracellular fluid, so the electrical potential extracellularly is going to go down because we're taking positive charge out of there. Okay, the positive charge was making the electrical potential higher. If we remove it, that's going to reduce the electrical potential. So, what effect is that going to overall have on the electrical potential difference? Well, initially, the intracellular compartment was negative 65 millivolts compared to the electrical potential extracellularly. But now this one's coming down, and this one's rising up to meet this one. So this number is going to become less negative, basically. Okay, right. So, basically, the fancy name for the electrical potential difference across the cell membrane becoming less negative is depolarization. Okay, so that's the process by which the electrical potential difference across the cell membrane becomes less negative. It becomes depolarized. Okay, right. And this will promote the formation of potentially action potentials within the cell if it's an excitable cell. Okay. If it's not an excitable cell, if it simply doesn't have the voltage-gated sodium channels and the voltage-gated potassium channels that you need to fire an action potential, then the change in electrical potential difference across the cell membrane may produce other changes. Okay, right, so ATP is going to induce depolarization of the electrical potential difference across the cell membrane by opening these P2X channels. Okay, right. Now, one more thing I want to just talk about is how you cancel the ATP signal, basically. Okay, so how is the ATP degraded? So let me just draw you a little cartoon of ATP. So basically, ATP stands for adenosine triphosphate, and I think I'll write out the name before I start drawing the cartoon. So adenosine, that's the A, and then the TP is for tri, and then phosphate. Okay, so in order to understand the structure of adenosine triphosphate, we need to know what adenosine is. Okay, so basically adenosine is the name for the organic base adenine. Okay, so here is the organic base adenine, which is being inserted by being just represented as a rectangle with an A written in it. Okay, so that rectangle now coloured in green with a letter A in represents the organic base adenine. And then we've bound it to the sugar ribose, which is this pentameric ring with a fifth carbon sticking off up here. Okay, so that's ribose. And basically when you have adenine bound to ribose, the combination of the two is then called adenosine. So ribose plus adenine is equal to adenosine. Okay, now, to convert adenosine into adenosine triphosphate, what you then do is you attach onto the fifth carbon of the ribose sugar within the adenosine 3 phosphate groups, like so. Okay, so this is 1, 2, and then 3. Okay, now these phosphates are labelled as such. The first one, which is attached to the fifth carbon, is called the alpha phosphate. The second one attached to the alpha phosphate is called the beta phosphate. The third one is then called the gamma phosphate. So, basically, enzymes which are on the outer surfaces of cells are going to start breaking down the ATP into first the ADP, adenosine diphosphate, and to do that what you do is you chop off the gamma phosphate. Okay, so then what you're left with is adenosine with 2 phosphate groups bound to it, and that's called adenosine diphosphate. Okay, but they won't stop there. They will then break off the beta phosphate off the alpha phosphate, okay, to leave adenosine with a single phosphate group now attached to it, which is adenosine monophosphate. Okay, and then finally they'll break off the alpha phosphate from the adenosine monophosphate to just leave adenosine. Okay, so then we'll just have adenosine. And then finally, the cell that originally secreted the ATP will then have a special transporter which can transport adenosine back into the cell. Okay, right, so that's how you terminate the signal, because only ATP can activate the P2X receptors, not ADP, AMP, or adenosine. Okay, so this is some sort of adenosine transporter here. Okay, right, so that's how you terminate the signal from ATP. Now, it can be used as a transmitter like this, but at other times it can signal cell damage, basically, because when cells are damaged, so let's say you've been punched and you've got cell damage, okay, they might lyse open, and then what they will release into the extracellular fluid is a lot of ATP, because they have a lot of ATP stored within their cytoplasm. Okay, this ATP can then go and work on P2X receptors that are on the surface of pain neurons, known as nociceptors, and will trigger action potentials in them by depolarizing the electrical potential difference across their cell membranes. Now, the two main P2X receptors that you have on the surface of nociceptors are P2X-free homotrimers and also P2X2-free heterotrimers. So let me just finish this and then we'll finish this video. So this is, and I'm really regretting writing that there, this is an adenosine transporter. Okay, right. So that now concludes our discussion of the P2X receptors.